Hi everyone. My name is Villoutreix. I'm going to present you the Embryo Digital Atlas, which is an open source project that I developed as a postdoc at Princeton University and a visiting fellow of the Center for Data Arts at the New School. So, as a researcher, I'm interested in embryogenesis, which is the drama of life before birth. So this is a very engaging cover that is 50 years old. And we still know very little of what is happening between the moment where the first cell is fertilized and you get a fully formed organism, and how every cell goes to the right position, how organs are forming, and how finally an organism is built. It's difficult to experiment with human embryos, so we are using model organism. The most famous one is the fruit fly, the Drosophila melanogaster. And to study it, we are using various techniques. A very recent one is light sheet microscope. So here you see the two sides of a 3D volume in time. And the nuclei have been stained, so you can see the position of each cell, how they are moving together as a tissue. And in this case, they are not dividing, but you can track how cells are proliferating. At the same time, you have dynamics of gene expression, so how proteins are positioned in space and how they interact. And usually this is a very reproducible process. And this is how cells differentiate, so how they acquire their fate. For example, if they will become nervous system or hair or muscles or bones. Closer to us, people are studying the zebrafish, which is another model organism. The difference is that it's a vertebrate embryo, so there are more mechanisms that are conserved between the humans and the zebrafish. This is kind of an old movie where you can see the development. So on top, you have the first cells developing here, 4, 8, 16. It's developing asynchronously. And below you can see the yolk, which is where the nutrients are. And in the beginning, the shape is very simple, and then it becomes more complicated. You have the spine that forms, and you will see the head appearing here, and the different segments, and the tail done there. With more recent microscopy technique, you can see it in 3D. So here it's a projection of a 3D stack in time, and again you see all the orange points are the nuclei, and then in green you have one protein in time, and here you see the head, which is the equivalent of this. And then another technique, which is optical tomography and also light sheet microscope, you can see at the same time the entire embryo and the cells of the nervous system in orange there. So to sum up, we can acquire many processes that happen in the development, and we try to understand how they are correlated and how they interact between each other to form an organism. And so there are dynamics at multiple scales, very short time and at a very short spatial scale, but at the same time at the level of the entire embryo. Similarly, we can use, in certain cases, this kind of microscopy on human embryos. So this is the leg of a human, and you see the nerves and the vascular system. But this is not a developing embryo, obviously. And this is looking at the foot, so you can see the arteries, and later on you will see the bone appear. So this is a very recent paper. It has been published last year. It's a bit slow. Yeah, and so in the end you can extract the bones from the foot. So we discussed it a lot this morning, but anatomical descriptions are work of art. This is an old drawing by Ramon y Cajal about the neurons, which is a still powerful way of engaging people into scientific work. His drawings has recently been published again in a book. This other one is Ernst Haeckel. He also drew many representation of many different species. So in that case, it's the Ascidian, but he grouped them by species. And Henry Gurry, which is also very famous and has inspired many artists, and he provided a very beautiful anatomical description of humans. So more recently, a visualizer provided this kind of visualization at the molecular level, where they synthesize scientific knowledge coming from different experiments by hand, and then they reproduce the 3D structure of a protein, or in that case of a virus, or the entire cell with the work of Gail McGill at DigiZyme. And here it's interactive, so you can, on the website, click on every molecule and have information about their functionality in the cell. So this is really beautiful and a very good tool for education. Joy was discussing it a little bit. The artistic bias is important to have a very interesting representation of the process, but at the same time, if the process is too complex, then maybe it will overcome the cognitive capabilities of the artist. So the question that arises from this is, can we produce this kind of integrative visualization in a data-driven way? So trying to remove as much as possible the bias of the person doing the visualization. The obvious way is to look directly at the data, and this is how databases of images in the case of development look like. So it's very difficult to, it doesn't look very accessible. It's very precise. You have a list of genes, a list of images with all the conditions for experiments. But we are very far from the representation of the molecular visualizer. So this is what I was working on in the past 2 years. Can we build accurate visualization of developing embryos that can be at the same time data-driven, interactive, and hopefully visually appealing? So for the data-driven part, when I was at Princeton, I had access to data which looked like this. So this is a cross-section of an embryo. On top, you have live imaging, similar to the first movie I showed you, where an embryo is developing in time, but you don't have access to the protein distribution because you cannot have it with the same technique. So then you have other experiments like those 4, where you can have partial view of the development at random time points. So in the end, you have an ensemble of images that all represent partial view of the same process, and you're interested in fusing them together to have an integrative view. So we derive some algorithm that does it using the morphology as a proxy for time. So it's published, and I can tell you more details, but in the end, we obtain a movie that animates all those snapshots of the different protein distribution. And from this, we can extract the position of the nuclei in time, and so have time courses of protein, and try to infer how the proteins are interacting, which one is the cause of the other, or if they are just correlation. So this was quite interesting to work on this, and we were using data that were already published. So it leads us to the question of how do we manage image data in developmental biology, and can we generate a platform where at the same time, can we generate a platform where we can visualize this kind of data? And this was the starting point for the project, the Embryo Digital Atlas, where the aims are twofold. On one side, it's for education and general access to the data, and on the other side would be a way to exchange data between researchers and have a way to publish data from labs and have them accessible for students. So this was done within the Center for Data Arts and also with the support of the Mozilla Science Lab. So Mozilla is a company that developed Firefox, which is an open source software, and they're trying to transfer all the practices of the open source development towards science. So it means open access publication, open source code, obviously, and open data. We developed the project on GitHub, which is a platform for open source development. And this is really nice because that way we could have people that don't have a PhD or background in science participate to a science project. So in this project, there is a list of issues up there. And I participated to a hackathon, and people from, for example, Chicago that I haven't met were telling me, oh, I'm interested, I know how to code in JavaScript, can I participate in your project? I said, yeah, of course. So in the end, yeah, we came out with a prototype for the website that you can see. It's online, and this is the movie I was showing you in the beginning. So you can toggle the different channels to see only the evolution of deep ear or twist, or only the morphology in time. You have also some info about the dataset. This is another one which is in 3D. You can interact with it. It's all in JavaScript. You can also toggle between, change the channel between membranes or nuclei. And for now, we have a list of 8 datasets that have been published, but the aim is to increase this and have it also together with some explanation in general of what is embryogenesis and cluster or aggregated into species or different developmental times. Yeah, and there is a description of the project. So the upcoming challenges of this is first, how do we give credit to people when they publish open data set. There are, for example, when you work on the fly, you will first produce a movie of the wild type development, and everyone does that in all the labs in the world. Could be just interesting to have a few of them that serve as references for everyone. And the limit is that usually people publish their data set only through publication, so when they try to show something, a very specific mechanism, whereas just a description of the organism could be useful for education. So there are some platforms where you can give a DOI for a data set. So we can use that. When we integrate several data sets together, it's also a question of how you give credit to the various data set that has been used when for the integrated view. I'm almost done. Yeah. So another aspect is can we have it in a collaborative way, like Wikipedia, where people can tag the data set so that you can teach each one can teach someone else about the data set. A project we just started is to transfer these 3D images into a VR headset. And finally, the question of colors. So the talk before just said that colors are important to engage people, and it's the case also when you have hundreds of colors mixed together. So with this, I would like to thank you, and this work was done with Stas Schwartzman at Princeton University, people from the Center for Data Arts, the Mozilla Science Lab, and I'm now working with the Weizmann Institute of Science. Thank you.